In this lecture, we'll look at diffusion of gases, blood flow to the lung, ventilation-perfusion relationships, and O2 and CO2 transport in the blood. In order to understand the movement of respiratory gases such as oxygen and CO2, we need to understand the concept of partial pressure. Partial pressure is the pressure exerted by a particular gas in a mixture of gases. So for example, as you can see in this figure here, we have gas A, the blue molecules here, and it exerts a partial pressure of P sub A. We have gas B, it exerts a pressure of P sub B, and gas C, it exerts a pressure of P sub C. So each of these gases exerts a pressure on its own independent of the other gases. So for example, with gas A, as these molecules hit the sides of this container, they exert a force. And the force that's perpendicular to a surface area is defined as being pressure. For example, when we talk about pounds per square inch, pounds is a unit of force, and the per square inch refers to the surface area that that force is acting on, that it's perpendicular to. In this case, we can combine all three. Imagine we have a box with all three of these gases in it. The total pressure then would be the sum of these partial pressures. So the total pressure, P sub total, is equal to P sub A plus P sub B plus P sub C. So this essentially shows you why these are called partial pressures. Each gas independently accounts for just a part of the total pressure exerted by a gas mixture. Dalton's law states that the total pressure of a gas mixture is equal to the sum of the pressure that each gas would exert independently. So if we take an example of calculating the partial pressures, the total pressure of a gas mixture such as air, and we'll assume that the air is dry, the total pressure of air is equal to the partial pressure of oxygen plus the partial pressure of CO2 plus the partial pressure of nitrogen. Let's look at the partial pressure of oxygen first. What's the partial pressure of oxygen in air at sea level? The total atmospheric pressure is 760 millimeters of mercury. Air is composed of 20.93% oxygen. Therefore, what is the partial pressure of oxygen, or PO2? To find the partial pressure of oxygen, what we need to do is convert the percentage to a fraction. So 20.93 would be converted to 0.2093. Multiply that then by the total atmospheric pressure, which is 760 millimeters of mercury. And we now have our partial pressure of oxygen, which is 159 millimeters of mercury. We can do the same calculation for CO2. It constitutes only 0.3%. Convert that to a fraction, it's 0.003. Multiply that by 760, and we have 0.3 millimeters of mercury. For nitrogen, it constitutes 79.04% of air. We can take 0.7904 multiplied by 760 millimeters of mercury, and we get 600.7 millimeters of mercury. I should point out that not all of what we're considering nitrogen is in fact nitrogen. About 78% of the atmosphere is nitrogen. There's another 1% that are noble gases such as argon. But because they're inert, we can actually lump them together with nitrogen in most situations. And therefore, from the perspective of respiratory physiology, we just consider those gases to be nitrogen. And that's how we get this 79.04% for nitrogen. As I mentioned earlier, there are 2 points in which diffusion become really important for respiratory physiology. The first is within the lung. Oxygen will have to diffuse from the alveolar airspace into the pulmonary capillaries, and CO2 will diffuse from the pulmonary capillaries into the alveolar airspace. The oxygen is then transported throughout the circulatory system. CO2 is also. Within the tissues, then diffusion becomes important again. Diffusion of oxygen goes in the direction from the systemic capillaries into interstitial space and then into the cells that make up a tissue. And CO2 would diffuse in the opposite direction. So we need to really understand how diffusion works. particularly across these slices of tissue, which we can refer to as tissue or as membranes. And the way we can do this is using Fick's law of diffusion. According to Fick's law of diffusion, the rate of gas transfer, or V gas, and this is a rate, it's the amount of gas per unit time, is proportional to the difference in the partial pressure of the gas on the two sides of the tissue, or as I said earlier, it could be considered sometimes as a membrane. So in this illustration, we can see we have gas at a certain concentration on one side of, say, a box, and we have gas at a lower concentration on the other side. Now we have a membrane here, or tissue, shown in red, and it's being held in place by these black blocks. We'll assume that they don't allow for the diffusion of gas at all. Assume they're metal or something like that. So these molecules, these green molecules, can diffuse through this membrane. Now, what would happen if we increase the concentration gradient? So we have a certain pressure, partial pressure, say P1 on the left side and P2 on the right side. What if we increase the partial pressure on the left side? So P1 is now greater, we're maintaining P2 constant. Well, now the pressure difference, or P1 minus P2, is greater, and this leads to a proportional increase in the rate of gas transfer. So V gas is proportional to P1 minus P2. So for example, if we make P1 minus P2 double, twice as big, then the rate of transfer of gas will also be doubled. According to Fick's law of diffusion, the rate of gas transfer, V gas, is also proportional to the tissue area or the membrane area. So for example, here I've drawn the tissue here in red. I've doubled the length. So let's imagine we've increased the area by a factor of 2. Now we would expect V gas to be twice as big. So the rate of gas transfer, the amount of gas that transfers from one side of this box to the other, would now be doubled. According to Fick's law of diffusion, the rate of gas transfer, V gas, is inversely proportional to the thickness of the tissue or the thickness of the membrane. So in this case, we have a membrane. It has a certain thickness. Everything is the same, except now the membrane is thinner. So if the membrane is thinner, that means that V gas will now be faster. There'll be a higher rate because we're now dividing by a smaller number. If we went in the opposite direction, if we made the membrane thicker, then that would decrease the V gas. So for example, let's take this is our starting point. Let's say we double the thickness. Well, now we'd be dividing by 2, and therefore the rate of gas transfer would be half of what it was originally. And this you can conceptualize as the fact that as these molecules go through this membrane, it's not just a simple straight shot through. It's almost like a little, imagine sort of a matrix with different passageways and so forth. And so as they go through, if you increase the thickness of the membrane, it simply becomes harder for the molecules to get through in a certain amount of time. And that increases linearly with the thickness of the membrane. And so this is important, as we'll see that later when we talk about this, that a lot of the membranes that we talk about when we need to have good rate of diffusion have to be very thin. According to Fick's law of diffusion, the rate of gas transfer, or V gas, is proportional to the diffusion coefficient of the gas. This is referred to as D. And so if there's an increase in this diffusion coefficient, then there'll be an increase in the rate of gas transfer. So for example, a doubling of D would lead to a doubling of the rate of gas transfer. Now, D is a little bit hard to explain because it incorporates various things. And typically, it's actually measured in an experiment, and then the values are put in a table. So the diffusion coefficient is dependent on the molecular size and other properties of the diffusing substance, as well as the properties of the tissue that the substance is diffusing through. And it's also temperature dependent. So, for example, an increase in temperature will generally lead to a higher D. And the reason for that is there's more thermal energy, the molecules are bouncing around more, and so they'll make their way through the tissue or the membrane faster. If we put everything together, we have Fick's law of diffusion. So V gas, which is the rate of gas transfer, is equal to the area divided by the thickness of the tissue, multiplied by the diffusion coefficient, multiplied by the difference in partial pressure. We can apply Fick's law of diffusion to the lung and the properties of the lung that increase diffusion rate. So first of all, within your lung, you have many alveoli, which are these tiny little sacs, and across them, oxygen will diffuse into the pulmonary capillaries, and CO2 will move from the pulmonary capillaries into the alveolar airspace. There are many of these tiny little alveoli, and so if you add up the surface area of all these tiny little sacs, that becomes a tremendously large surface area. So according to Fick's law of diffusion, an increase in surface area should lead to an increase in the rate of diffusion, and that's what occurs in your lungs. The other thing that happens is your alveolar membrane is very thin. So these are squamous epithelial cells, which means they're very flattened. They have a fused basement membrane with the endothelial cells of the pulmonary capillaries. So essentially, oxygen would have to go through a membrane, another membrane, through a small basement membrane, and then two more membranes to get from the pulmonary from the alveolar airspace into the pulmonary capillaries. And that's a very short distance, and so it diffuses quite easily. Through ventilation, which means as we're breathing in and out tidally, we refresh the air in our lungs. So as we increase the oxygen level, we maintain the partial pressure of oxygen relatively high in the lungs in this alveolar airspace in comparison to the pressure within of PO2 within the deoxygenated blood that's coming from the right ventricle. And so this large pressure difference, partial pressure difference for oxygen, leads to rapid diffusion of oxygen. In fact, by the time it leaves the lungs, the oxygen level is very high. And likewise, we can talk about CO2. There's also a partial pressure difference for CO2, such that it's higher within the blood plasma of the pulmonary capillaries relative to the alveolar airspace. And again, the reason for this is because we're ventilating, we're breathing in and out, and so we maintain the level of CO2, the partial pressure of CO2 within the alveolar airspace lower than that of within the pulmonary capillaries. There are two general rules to keep in mind. Gases always move down their partial pressure gradient, and then also each gas moves independently of the other gases. Let's take the example of oxygen. As blood goes through the pulmonary artery to the lungs, it has a partial pressure of oxygen, or PO2, of 40 millimeters of mercury. Within the alveolar airspace, the partial pressure of oxygen is 104 millimeters of mercury. By the time the blood leaves in the pulmonary veins, coming back to the left atrium, it's now at 95 millimeters of mercury, so it's very high. It's almost as close as very close to the level of oxygenation within the alveolar airspace. We can also look at CO2. It's at 46 millimeters of mercury in the pulmonary arteries. If we look within the alveolar airspace, it's 40 millimeters of mercury, so note that that's lower than the oxygen within the blood, and therefore there's a loss of CO2 by diffusion. It's exhaled, and then within the pulmonary veins, coming back to the left atrium, we have a PCO2 of 40 millimeters of mercury. Note that that's the same as the PCO2 within the alveolar airspace. The take-home messages are that gas moves from the blood across the blood-gas interface in the lung due to simple diffusion. Recall that simple diffusion is diffusion directly across a lipid bilayer without the aid of a protein transporter. The rate of diffusion is described by Fick's law, and diffusion is greater when the surface area is large, when the tissue thickness is small, and when the driving pressure or difference in partial pressures is high. The pulmonary circuit goes to the lungs, whereas the systemic circuit goes to the rest of the body. So since blood is moving around in a circle effectively throughout the body, this means that the flow rate through the pulmonary circuit is exactly the same as the flow rate within the systemic circuit. And by flow rate, I mean the volume of blood that passes past a certain point per unit time. Therefore, for example, if you were to look at the pulmonary trunk and you would measure how much blood flows through that per minute, it would be about 5 liters per minute for a typical adult. And that would be exactly the same as the flow rate moving from the left ventricle out into the systemic circuit through the aorta. Again, you'd see 5 liters per minute for this typical adult. Okay, so the flow rate is the same. However, the pressures are very different. The mean arterial pressure for the systemic circuit is about 100 millimeters of mercury. The mean pressure for the pulmonary circuits is much lower. And why is it lower? Well, we don't really need as much driving force to move blood through the pulmonary circuit. You can see here in this diagram, the pulmonary circuit's much shorter. It's much simpler. Furthermore, really high pressures would damage the very delicate structures, such as these very fine pulmonary capillaries and so forth that we just described. And so we really don't need a very high pressure, and in fact, we don't have a very high pressure within the pulmonary circuit. So how do we increase blood flow during exercise? Well, during exercise, resistance falls due to vasodilation, and also due to the opening of unused capillaries, which we'll talk about a little bit more in the future. But this is called recruitment. This is called recruitment, when we start to open up some unused capillaries, pulmonary capillaries within the lungs. So this is how we get more blood flow. We essentially have a vasodilation and opening of unused capillaries, and this allows for higher blood flow without much of an increase in the pulmonary pressure, even when we're exercising. Recall that your blood pressure isn't constant throughout your body. In other words, if you were to measure the mean arterial pressure within the aorta, it's about 100 millimeters of mercury. And you can measure this using a blood pressure cuff on the brachial artery within your upper arm. That's about at the same level roughly as the heart. And so you get a pretty accurate measure of the aortic pressure there. However, if you were to measure your blood pressure, say in your foot when you're standing, well, it actually would be 90 millimeters of mercury higher in an artery in your foot than in your aorta. And the reason for that is gravity. So there's essentially a column of fluid, and gravity acting on that fluid will cause an increase in pressure. Of course, if you're lying down, then that effect would go away. So your lungs exhibit a similar phenomenon. So your lungs, in an upright human, there's about 30, adult human, there's about 30 centimeters difference between the highest point and the lowest point within your lungs. So this turns into about a 23 millimeter pressure difference, with the pressure being higher at the bottom of the lung and lowest at the apex of the lung. And so this actually has a pretty significant effect on blood flow through the lung. So if you think about it, within the lung, you have these pulmonary capillaries, but you also have the alveoli. And within the alveoli, there are little tiny sacs, and within each of them, there's an air space, and that air has a certain pressure. Now, at the top of the lung, it's actually possible, as you can see here in this diagram here, for the pressure within the alveoli to be greater than that of the pressure within a pulmonary capillary. And so this acts like a lot like a pressure cuff when you're measuring someone's blood pressure. It squeezes the blood vessel, the capillary, and it can greatly reduce the blood flow through the capillary. Now, this effect is going to be a lot more prevalent at the top or apex of the lung than it would be at the bottom. At the bottom of the lung, the arterial pressure is 23 millimeters of mercury higher than it is at the top, and so that's sufficient to open up the capillaries. In other words, the pressure is greater than that of within the alveolar airspace, and therefore there's no impedance. Nothing impedes the flow of blood through the capillaries. And so the overall takeaway message is if you look at the lungs, there are differences in regional blood flow within the lung. So here we have blood flow on the y-axis. On the bottom here, we have the bottom of the lung represented here and the top of the lung up here, and we have different rib numbers. So as we go from the bottom of the lung towards the top, the blood flow rate, again, a volume per unit time, it decreases by the time we get to the top of the lungs. Now, of course, this effect, if you're lying down, will go away. The blood flow or blood perfusion of the lung would be more or less uniform throughout the lungs. Now, when you exercise, what happens is you do have an increase, a little bit of an increase in the pulmonary arterial pulmonary pressure, and therefore you do get more blood flow than at the apex. And this is what I talked about previously. You'll get more recruitment of more blood vessels during exercise. And so some of the blood vessels that were had very slow rates of blood flow, or may even have been constricted enough so that there was no blood flow at all, these will open up, and now you'll have more blood flow through more capillaries as you're exercising. The take-home messages are that the pulmonary circulation is a low-pressure system with a rate of blood flow equal to that of the systemic circuit. In a standing position, most of the blood flow to the lung is distributed to the base of the lung due to gravitational force. Previously, I mentioned that due to the effect of gravity, there's more blood flow through the base of the lungs relative to the apex of the lungs. Now, it turns out that that leads to what can be referred to as a ventilation-perfusion mismatch. So ventilation is defined as the volume of air that reaches the alveoli, and perfusion, and that's represented with V, and Q represents perfusion. So perfusion is the blood that reaches the capillaries that go right past the alveoli. So ideally, you'd have about, it works out such that for about 1 liter of air, you should have about 1 liter of blood. And that's very close to the ideal perfusion rate match, ventilation-perfusion ratio. And so that would indicate very good matching. It turns out if you look at the lung overall, the value is about 0.8, which means it has slightly more perfusion, which is Q, relative to ventilation, which is V. But if we look at specific parts of the lung, the story is even more extreme. So let's look at the apex of the lung. Ventilation is 0.24 liters per minute. Blood flow through the pulmonary capillaries in the apex is only 0.07. So if you take 0.24 and divide it by 0.07, you get 3.43. So this is the ventilation-perfusion ratio, and that's very far from the ideal value of roughly about 1. And so this indicates that given this amount of ventilation, there's not nearly enough perfusion. It should be ideally about 0.24. Now, if we look at the base of the heart, the situation is a bit different. We have a certain amount of ventilation, 0.82 liters per minute. The blood flow is 1.29 liters per minute. And again, we take ventilation divided by perfusion, and here we get 0.64. So this is actually in the opposite direction, and it's now below the ideal value of 1. So this means there's actually more blood flow than is really necessary. And so if we look at the apex of the lung, it's underperfused because it has a ratio of less than 1. And the base of the lung is overperfused relative to the amount of ventilation because it has a ratio of more than 1. And as I said, overall, if you're at rest, the ventilation-perfusion ratio is about 0.8. So it's not ideal. And that would correspond to a little bit more perfusion relative to ventilation. Now, the situation actually gets better. If someone's doing light exercise, that actually improves the ventilation-perfusion ratio. And the reason for this is clear from what we discussed earlier. As you exercise more, there's a higher pressure in the pulmonary circuit, and that tends to open up some of the pulmonary capillaries that are higher up in the lung, getting from, halfway, the middle third, they're going to start to open up, also even in the apex. So that would actually tend to increase the amount of blood flow, and that would help with this ventilation-perfusion ratio and bring it closer to 1. Now, it turns out actually, when really heavy exercise, this actually, again, results in a ventilation-perfusion inequality. And it's not exactly clear why that occurs, but it turns out that the best ventilation-perfusion value is actually found with more light or moderate exercise. The take-home message is that efficient gas exchange between the blood and the lung requires proper matching of blood flow to ventilation. This is referred to as ventilation-perfusion relationships. It turns out if you do the calculations, the ideal ratio for ventilation to perfusion is about 1, meaning that for, say, 1 liter of air reaching the alveolar airspace, there should be about 1 liter of blood that passes through the pulmonary capillaries that are associated with those alveoli. And this ratio would imply a near perfect matching of blood flow to ventilation. Compared to carbon dioxide, oxygen actually has a relatively low solubility in water. Because of this, very little of the oxygen that's transported to your tissues is actually transported simply dissolved in the aqueous part of your blood, in the blood plasma. 99% of the oxygen is transported bound to hemoglobin. There are 2 forms of hemoglobin: oxyhemoglobin, which is hemoglobin bound to oxygen, and deoxyhemoglobin, which is hemoglobin that's not bound to oxygen. The amount of oxygen that can be transported per unit volume of blood is dependent on the hemoglobin concentration. For each gram of hemoglobin, 1.34 milliliters of oxygen can be transported. So we can do some calculations. If we just make some simple assumptions, let's assume that blood is 100%, has hemoglobin that's 100% saturated with oxygen. This means that it can't carry any more oxygen bound to hemoglobin. If we took a male, a typical male has 150 grams of hemoglobin per liter of blood. If we multiply that by this conversion factor, which is for each gram of hemoglobin, we have 1.34 milliliters of oxygen that's transported. You'll notice here that the grams of hemoglobin here is in the numerator. Here it's in the denominator, so that cancels out, and we'll have units of milliliters of oxygen in the numerator and per liter of blood in the denominator. And so our answer is 200 milliliters of oxygen per liter of blood. Now, typical females have a lower amount of hemoglobin per liter of blood. Recall that females have a lower hematocrit compared to males. And so we can do a similar calculation, and we find that females can carry 174 milliliters of oxygen per liter of blood. Oxygen can bind to 4 different sites per hemoglobin molecule. There are 4 globular subunits to hemoglobin. There are 2 alpha chains and 2 beta chains. And each of these globular proteins, or subunits, I should say, has a heme group, which is a prosthetic group that has iron in the middle. And oxygen is able to bind at that iron site. So per molecule of hemoglobin, you can actually have different levels of saturation of oxygen within just a single molecule. You could have no oxygen bound, you could have 1 molecule of oxygen, you could have 2 molecules, you could have 3. If all 4 sites are bound, then you would have 4 molecules of oxygen bound to a single molecule of hemoglobin. It turns out that the molecular structure of hemoglobin with the 4 sites that are able to bind oxygen gives rise to a very interesting curve when you look at percent oxyhemoglobin saturation overall. So imagine you have a beaker with hemoglobin in it. You could measure, and you had a device to measure how much of that hemoglobin was saturated. One possibility is every single site on every single hemoglobin molecule is saturated with oxygen. This would yield a value of 100% saturation. The other possibility is none of the sites are bound with oxygen, and therefore you'd have a 0% saturation. So this in fact can happen. So for example, if you have a high level partial pressure of oxygen, meaning there's a lot of oxygen within the fluid, within the beaker, then you can essentially have about 100% saturation. And then on the opposite extreme, let's say you bubble nitrogen gas through the beaker so that that displaces all the oxygen. So there's no oxygen whatsoever in the beaker. At that point, all the oxygen would have come off because it's not bound covalently. It's bound in a looser fashion than a covalent bond. So the oxygen can dissociate from the hemoglobin. And so then you'd have a 0% saturation value. Now, what's interesting, of course, is that you can have intermediate values. And this you get what is called a sigmoidal curve because it's roughly S-shaped. If you look at this, it's sort of S-shaped. And the nature of the curve actually has to do with the molecular structure. So it shows what's called cooperativity. So initially, to bind the first molecule of oxygen is a little bit difficult, but then once it binds one molecule of oxygen, that leads to a conformational change at the other three sites, and it becomes a little more likely to bind oxygen. And once, say, two sites are bound, it's even more likely to bind a third and then a fourth oxygen. Now, eventually, this sort of plateaus because eventually, at some point, you've got many of the sites are bound with oxygen, but since oxygen can also dissociate, you don't get to a full 100% saturation until you have a high level of oxygen in the liquid. Okay, so basically, and this idea, of course, is based on whatever we discussed, which is you have deoxyhemoglobin, which can bind reversibly with oxygen. When it binds to oxygen, it forms oxyhemoglobin. But then again, this isn't a covalent bond, and so the oxygen can dissociate from the oxyhemoglobin, and you can go back to having deoxyhemoglobin and oxygen within the solution. Let's next look at what would happen within the human body. In the lung, you have an alveolar airspace that has a high partial pressure of oxygen. That oxygen can diffuse into the pulmonary capillaries and give rise to a high partial pressure of oxygen there. That would then lead to saturation, nearly 100% saturation of the hemoglobin traveling within the red blood cells within the pulmonary capillaries. So this phenomenon is referred to as loading. The oxygen is being loaded up onto the hemoglobin within the red blood cells. Next thing that happens is the blood returns to the left atrium, then goes to left ventricle, then to the aorta, and out to the systemic circulation. It then passes through the arteries, getting to the arterioles, and then to the capillaries. Within the capillaries, within tissue, you'll have unloading. And the reason for this is because the tissue within the body has a lower partial pressure of oxygen within the cells and within the interstitial space. So this leads to diffusion of oxygen from the blood plasma into the interstitial space and into the cells. And therefore, we get an unloading phenomenon in which oxygen is leaving the hemoglobin within the red blood cells and entering into the tissue and being used up for aerobic respiration. And when a person's at rest, this phenomenon is called unloading. And when a person's at rest, the veins at rest would have a partial pressure of about 40 millimeters of mercury by the time the blood has exited the capillaries and is being collected in the venules and going into small veins and so forth. I should point out that this graph actually has two y-axes. One on the left shows the percent saturation of the oxyhemoglobin. The other axis, which is on the right, shown here, is showing the oxygen content. Oxygen content is the amount of oxygen, often expressed as a volume of oxygen, per volume of blood. So for example, here it's milliliters of O2 per 100 milliliters of blood. And what you notice here is that when you have 100% saturation, you have about 20 milliliters of oxygen per 100 milliliters of blood. That's at 100% saturation. By the time the blood is passed through the capillary beds and is being collected in veins, the oxygen content is now 15 milliliters of oxygen per 100 milliliters of blood. And so this gives us some indication that oxygen has been unloaded to the tissues. So 5 milliliters of oxygen per 100 milliliters of blood has potentially is unloaded. And you can combine this then with for example, the blood flow, a volume per unit time, to give you a sense of how much oxygen is being unloaded per unit time. So far, we've looked at the effect of PO2 on loading and unloading of oxygen from hemoglobin. It turns out there are other factors besides PO2 that affect this process of loading and unloading. And one thing that's very important is the affinity between hemoglobin and oxygen. It turns out that that's not constant. There are many things that can affect this affinity, which we'll go into next. One of the major factors that affects the affinity of hemoglobin for oxygen is pH. Let's take a look at this graph. At a standard pH for arterial blood, which is 7.4, you have this sigmoidal curve. Now, if there's something that causes an increase in hydrogen ion concentration in the blood, that would lead to a decrease in pH. And that shifts this whole curve to this new curve here. And this is referred to as a rightward shift because, as you can see, the curve is shifting and moving over to the right. And so this is actually helpful from a physiological point of view. When, say, someone's exercising, this leads to the generation of CO2. An increase in CO2 level in the blood can lead to a increase in hydrogen ion concentration, therefore a decrease in pH. If a person actually gets to the point where they're doing anaerobic respiration, that leads to the generation of lactic acid. Again, the hydrogen ion concentration would go up in the blood and the pH would go down. And these effects tend to be very somewhat localized also to the tissue that's actually very active. And so within active tissue, you might find the pH decreasing within the interstitial fluid. And this would then affect the pH within the capillaries, feeding that particular tissue that was very active. And so this actually helps with offloading. And let's see how that works. Well, let's imagine that we're at 40 millimeters of mercury. Under standard pH conditions, that would mean that our percent saturation is 70%, okay? So that means that there could be some offloading of oxygen from the hemoglobin, but there's still quite a bit of oxyhemoglobin. And there's still, in other words, there's still a lot of oxygen that's bound to hemoglobin. But with this rightward shift, let's go from 40 millimeters of mercury. Now we're roughly at 58 or something like that percent saturation. So there's a lower percent saturation. This means that more of the oxygen has dissociated from the hemoglobin and less is still bound to the hemoglobin. Instead of 70% bound, it's now 58%. So this favors the idea of offloading. You're getting rid of more oxygen, which is exactly what you need for tissue that's very active. This phenomenon is referred to as the Bohr effect. The pH dependency of this curve was first discovered by Bohr. Now, if you go in the opposite direction, let's say the pH increases, meaning there's a decrease in hydrogen ion concentration. Let's again do 40 millimeters of mercury. Instead of 70%, now we have 80% saturation. And if you're trying to unload oxygen, that would be a bad thing. That would mean that less oxygen is being unloaded because more of the oxygen is still bound to hemoglobin. Here I've just simply added the terms rightward and leftward shift to make it even clearer. So when you have a decrease in pH, you have a rightward shift right here with this curve here. And when you have an increase in pH, you have a leftward shift with this curve here. Another factor that has a large impact on the percent oxyhemoglobin saturation is temperature. So the standard core temperature for a person is 37 degrees. If the core temperature increases or the blood temperature increases, let's say to 42 degrees, then there's a rightward shift in the curve. So this would have the same effect that as we discussed with low pH, which is that there'll be enhanced offloading of oxygen to tissue. And again, this is actually helpful. Skeletal muscle that's very active will have a high temperature because of friction and because of the utilization of ATP, which is not 100% efficient and so forth. So there's a lot of reasons why there would be a high temperature in very active tissue. And that tissue would need more oxygen than less active tissue. And so this offloading phenomenon is enhanced by this increased temperature, which is helpful. And notice you can go in the other direction. If you have a lower than normal temperature, then this actually means that, let's start at 40 millimeters of mercury, that you would have more saturated hemoglobin, meaning that the hemoglobin and oxygen have a high affinity for one another, and there's a lot of it still bound to one another. There's another factor that has a large impact on the oxygen saturation percentage of hemoglobin, which is 2,3 DPG, which stands for 2,3-disphosphoglyceric acid, which is also referred to as 2,3-bisphosphoglyceric acid. So red blood cells don't have nuclear mitochondria, therefore they produce ATP by glycolysis only. A byproduct of glycolysis in red blood cells is 2,3-DPG. So it turns out 2,3-DPG can bind to hemoglobin, and it reduces hemoglobin's affinity for oxygen. So this leads to, just like high temperature and low pH, this leads to a rightward shift in the sigmoidal curve. As you can see here, they mentioned, for example, an increase in 2,3-DPG leads to a rightward shift. So we shift it over this way. And this would favor unloading of oxygen in tissue. So it turns out this is very interesting because if a person is at low altitude and then they go to high altitude, within a few days to weeks, they're going to start to increase the concentration of 2,3-DPG within the red blood cells. And this is just something that happens naturally as a consequence of being exposed to essentially hypoxic conditions where you're not getting as much oxygen as you normally would. And so this actually favors unloading of oxygen at tissues. And so that is actually helpful. It helps because, again, when you're at high altitude, the amount of oxygen, the partial pressure of oxygen in the atmosphere is lower. And therefore, it's actually harder to get enough oxygen to your tissues. And so this enhanced unloading due to the increase in 2,3-DPG is very helpful when someone's moves to a high altitude environment. We've talked a lot about hemoglobin, which is an oxygen binding protein. It turns out there's another very important oxygen binding protein, which is found in skeletal muscle as well as cardiac muscle, and this is myoglobin. And it's found particularly in large quantities in slow twitch fibers. These are the oxidative fibers, the ones that are very fatigue resistant. So for example, in your body, postural muscles that help you sit upright and stand upright and so forth tend to be ones that are on all the time, and they are very efficient, and they use oxidative mechanisms. In other words, aerobic metabolism. Because of that, they need a lot of oxygen. And it turns out, and we'll talk about this more in the next slide or so, that myoglobin is helpful for getting oxygen from the blood into the cells. Well, it serves as a sort of storage point for the oxygen within cells, which then helps to feed the oxygen to the mitochondria. Now, if we look at various different types of muscle, you'll see that they vary in color. And a lot of this color actually has to do with myoglobin. There are some other factors too, but myoglobin is one of the major reasons why muscle has different colors. So for example, fish, certain white fish like flounder, have very little myoglobin, so or none, and so they have a whitish color. Chicken is intermediate; it has quite a bit of myoglobin. And if you look at chicken, there's differences between different tissue within chicken. So breast meat is quite white and pale, and so it has relatively, and the reason is because it has relatively little myoglobin. Whereas the other muscles, for example, the thighs and legs, have a lot more myoglobin. They'll have a darker color to them. And then finally, if you look at, say, something like a cow, its meat has a lot of myoglobin, particularly the meat that comes, the muscle that is involved, again, in slow twitch, has a lot of high percentage of slow twitch fibers that have a lot of oxidative metabolism. I should quickly mention that smooth muscle can also have myoglobin. One example of this is the gizzard in the chicken. So this is smooth muscle that's involved in grinding food. It has a very dark color, and the reason for that is it has a lot of myoglobin. So smooth muscle also can have myoglobin. This figure shows a comparison of the saturation curve for hemoglobin and the saturation curve for myoglobin. You can see that the hemoglobin shows this characteristic S-shaped curve or sigmoidal curve, whereas the myoglobin doesn't really show that. It shows a simpler curve. And the reason for this, myoglobin is actually a little bit simpler molecularly. But the main important thing to notice here is that the affinity of myoglobin for oxygen is much higher than that of hemoglobin. Let's again look at 40 millimeters of mercury, which would be the average value for the partial pressure of oxygen within venous blood. So for hemoglobin, you would have about 75% saturation, meaning that the hemoglobin would tend to give up at least some of its oxygen to the surrounding aqueous medium around it. Now, for myoglobin, at that same value of 40, you have closer to maybe 90% saturation. So that means that at a given partial pressure of oxygen, there's a much higher affinity for oxygen between oxygen and myoglobin than between oxygen and hemoglobin. Now, how is this useful? Well, so it turns out that as the blood passes through the capillaries and it has oxygen in it, the oxygen does dissociate from the hemoglobin. It then moves by simple diffusion through the endothelium of the capillaries into the interstitial space and then into cells. And eventually it's going to go into mitochondria, where it essentially disappears as it turns into water, as it accepts an electron and hydrogen. Now, the thing is, you need to get the oxygen in, so it needs to be passed, and it could simply go directly into the mitochondria without myoglobin. But myoglobin is helpful because it serves as sort of an extra storage stage for the oxygen. The oxygen can come in, it binds to myoglobin, and it only will dissociate when the oxygen level drops, a bit lower. And it does drop a little bit lower. So in active skeletal muscle, the partial pressure of oxygen can be very low. And in fact, right around the mitochondria would be even lower. So there's even gradients within the cell itself. And so in this way, myoglobin sort of temporarily stores the oxygen, but then it will give it up to the mitochondria, which then use it up for aerobic respiration. So it's sort of, you can think of it as the hemoglobin sort of passing the oxygen to the myoglobin. It holds on to it for a very brief period of time, and then it passes the oxygen on to the mitochondria. So what is the evidence for this? Well, there's a couple lines of evidence, and one that the myoglobin serves as a temporary storage of point for oxygen. One is this idea that when you start exercise at the very beginning, your muscles are very active, your skeletal muscles are very active. They use up a lot of oxygen to generate ATP aerobically, and yet your cardiopulmonary system always has a little bit of a delay, so you're not quite up to speed in terms of getting enough oxygen delivered through the cardiovascular system. And so the myoglobin at least seems like a very logical place where you're storing just enough oxygen to start up that process, and that oxygen can be fed into the mitochondria and therefore used for aerobic respiration. And eventually, as you bring up the delivery of oxygen to the tissue, you can actually start to restore some of that oxygen to the myoglobin. And when you stop the exercise, that's actually going to be part of the excess ventilation and so forth that occurs at the end of exercise, and that helps to restore the oxygen to the myoglobin if it had been depleted at all. So at least that conceptually makes a lot of sense, that myoglobin would have this sort of temporary storage effect. But then there's even stronger proof that comes from looking at different types of animals. So as I mentioned earlier, we can look at different muscle and we can get a good idea of how much myoglobin there is just by looking at its appearance. Of course, there's tests that can give us the exact amount of myoglobin per se kilogram of muscle. So it turns out that diving animals, such as whales, have so much myoglobin, their muscles actually look black. Here's a close-up of a Weddell seal mussel. The color of its muscle is nearly black. And people, of course, studied this, and they found that there's, in fact, just a tremendous amount of myoglobin. In fact, there's so much myoglobin that for a while it was a mystery. They didn't understand why the myoglobin didn't just sort of stick together. All this protein would stick together and precipitate out. And then there's been recent studies showing that the myoglobin is actually positively charged. And so the part of the reason they don't think it precipitates out and becoming solid clumps is because it tends to repulse. It's one molecule of myoglobin will repulse another molecule because it's like trying to push two magnets together that have the same poles. These positive ions tend to push away each other. So far, we've looked at the transport of oxygen throughout the cardiovascular system. Well, equally important is the transport of CO2. As you know, as our cells respire aerobically, they generate a lot of CO2 as a metabolic byproduct. So CO2 is much more soluble in an aqueous solution compared to oxygen. That's why CO2 is used to make soda fizzy, for example. And so CO2 is dissolved in plasma, accounts for about 10% of the transport of CO2. So we can look at this picture here. CO2 is produced in the cell. You have dissolved CO2 in the interstitial space. It can pass through cell membranes without any trouble by simple diffusion as a gas. And then it can travel just as CO2 in the solution within the blood plasma. Now, 20% is bound to hemoglobin. So if we look at this diagram here, some of the CO2 can enter into the red blood cells. Within the red blood cells, there's hemoglobin. And the hemoglobin can actually bind CO2. It's not at the same site. It's at another site within the globular subunits, which are, of course, chains of amino acids. Finally, the majority of CO2 is transported in your blood in the form of bicarbonate. In tissue, let's take a look at this. So CO2 is produced, dissolves, it then enters, is able to enter into a red blood cell, and CO2 and water can combine to form carbonic acid. And the Ca stands for carbonic anhydrase, which is an enzyme which tremendously speeds up this reaction. So it helps in the formation of carbonic acid. Now, carbonic acid will spontaneously break apart into bicarbonate, which is HCO3 minus, and hydrogen ion. So the next step is that this hydrogen ion would be problematic, right? But it can actually bind to hemoglobin also. And so the hemoglobin acts as a bit of a buffer. So it's helping. So this would cause, obviously, an increase in hydrogen ion concentration would decrease the pH, meaning it would become acidic within the red blood cell. But the hemoglobin itself acts as a buffer, so it allows hydrogen to bind to it. So that takes care of the hydrogen. So now what's happening though is we have the buildup of bicarbonate. Now bicarbonate has a negative charge, so it can't leave the cell very easily without causing an ion imbalance. So bicarbonate will be transported. Some of it will be in the red blood cells, but a lot of it will be in your blood plasma. So how do we get the bicarbonate out? Well, it can move out. So there are ways for it to get across. There are channels in the membrane that allow for this ion to leave down its concentration gradient. But the problem is that if a negative ion leaves, that would lead to there being a charge imbalance. There would be more positive ions within the cell compared to outside. And so what happens is there are also chloride channels, and chloride can go down its concentration gradient from outside to inside, and that helps to balance this balance the charges across the membrane of the red blood cell. And so this is referred to the chloride shift. As bicarbonate leaves, these negative ions leaving would cause a problem, and so chloride enters, and that tends to keep the cell from becoming out of electrical balance. This slide simply shows the reaction a bit better. So again, bicarbonate is the form in which most CO2 is traveling in your blood. So 70% is traveling in the form of bicarbonate. And so CO2 can combine with water, forming carbonic acid. And again, carbonic anhydrase is the enzyme that speeds that reaction up tremendously. It can go forward, but it would just be very slow without the enzyme. And then carbonic acid can break apart quite quickly into hydrogen ion and bicarbonate. Next, the blood is transported to the pulmonary capillaries, and everything we talked about is essentially reversed. So recall that you have 10% of CO2 simply dissolved in the blood plasma. That can move down its concentration gradient into the alveolus and then is expired. Now, as this drop in CO2 within the blood plasma, because of it diffusing down its concentration gradient, essentially triggers a bunch of other things to happen. So, for example, recall that some CO2 was bound to hemoglobin. Well, these can dissociate, and again, the CO2 can exit and then leave the body as it's expired. A decrease in CO2 here within the cell also has an effect. Recall that there was the enzyme carbonic anhydrase within red blood cells. Well, that can help in the formation of carbonic acid from CO2 and water. But if there's a reduction in the amount of CO2, carbonic anhydrase will actually help the reaction go in the direction of breaking apart carbonic acid and making water and CO2. So again, we have CO2, which can exit the body. So a decrease in carbonic acid will have the effect of driving more formation of carbonic acid from bicarbonate and hydrogen ion. Recall that some of the hydrogen ion was able to bind with hemoglobin. So it can unbind and then form together with the bicarbonate more carbonic acid. And so as this bicarbonate drops in concentration within the cell, more bicarbonate can enter. And at the same time, recall that we had a lot of chloride in the cell. And so that chloride can now exit the cell. And so essentially the chloride shift in the opposite direction. So basically that's it. Everything is reversed, and the overall effect of all these changes is that CO2 leaves the blood plasma within the pulmonary capillary, ends up in the alveolar airspace, and then as you breathe out, it leaves the body. The take-home messages are more than 99% of oxygen is transported in the blood, chemically bonded with hemoglobin. The effect of partial pressure of oxygen on the combination of hemoglobin and oxygen is illustrated by the S-shaped oxygen hemoglobin saturation curve. An increase in body temperature and a reduction in pH results in a right shift in the oxygen hemoglobin dissociation curve and a reduced affinity of hemoglobin for oxygen. Carbon dioxide is transported in blood in three forms. One, dissolved CO2, that means about 10% of CO2 is transported in this way. Two, CO2 is bound to hemoglobin called carbaminohemoglobin. About 20% of blood CO2 is transported via this form. And three, bicarbonate. 70% of CO2 found in the blood is transported as bicarbonate or HCO3-.